Hi there, this is the video for review for midterm 2. It will be organized like the video for the first midterm. I've taken all the slides from all the presentations this part of the course. I've kept only the most important slides, and I will go through them one by one, telling you the main point about each slide. I won't be going them into comprehensive detail, but it is this main point that you are most responsible for on the test. Like last time, 95% of the test will come from this video. But this time there are at least two changes. First off, I want to congratulate everybody on the job they did on midterm 1. It was a great performance. This time the midterm will be more challenging in order to get a greater grade distribution spread so that people can distinguish themselves more easily. Secondly, the essay questions on midterm 1 didn't work out quite so well. You'll recall on that test we gave 10 terms that you were supposed to stitch together into an essay about a process like protein translation. Instead, what we found is we would get 10 unrelated sentences that didn't convey clear understanding of the process. So in this case, we will not give the 10 terms that you're supposed to use, but we'll still ask you to describe a process in order to help you prepare for the essay questions which are worth the most points. I list here on this slide some of the major topics that we've covered this portion of the course. And since these are the major topics, you might expect them to be represented at a high rate on the questions that are worth more points on the test. In the first lectures, this portion, we talked about extraction of energy from food. We talked about how there are three main components of this: glycolysis, which is breaking down glucose into pyruvate; the citric acid cycle, which is converting acetyl-CoA into high-energy electrons in NADH; and then the third process, which was on the second lecture, was oxidative phosphorylation, how these high-energy electrons are converted into ATP. If we start with the highest level view of metabolism, we know that it has two main functions: one, to provide energy for cellular processes, and two, to provide the molecular building blocks for needed biochemicals for the cell. So in its highest view, we have sugar or carbohydrate being oxidized down into carbon dioxide. As we're oxidizing these molecules, we're producing ATP. ATP and synthetic intermediates can be combined into the synthesis of amino acids, nucleic acids, and lipids. Here, showing the glycolysis and the citric acid cycle, some of the branch points for various of these compounds: nucleotides, lipids, and amino acids. You're not responsible for the specific molecules made from various intermediates, but just you need to be aware of the dual functions of metabolism. There are some limits to our ability to perform biosynthesis, and there are 10 amino acids that we cannot synthesize from sugar. These we need to obtain via protein in our diet. And we pointed out that it's curious that humans have lost this ability, but many so-called lower organisms, even bacteria, have no problems producing these 10 amino acids, all 20 of these amino acids from simple sugar inputs. But you're not responsible for the names of these 10 amino acids as being essential or non-essential. This is a high-level view of the digestion process. The food that we eat is digested in the mouth and the stomach into constituent parts: amino acids, sugars, and fatty acids. These are taken up into cells. In cells, the sugars are in the cytosol, are converted from glucose to pyruvate, and in the mitochondria, acetyl-CoA is converted to carbon dioxide, producing ATP. This process is dependent on oxygen as a final electron acceptor, and CO2 is exhaled as part of the waste removal. Food that we eat are stored in two forms: glycogen and fatty acids in adipose tissue. Fats are stored in the form of triglycerides, which is 3 fatty acids linked by a glycerol backbone, and glycogen in the liver and muscle, which are branched polysaccharides of glucose molecules and other sugars, which can then be hydrolyzed here at this bond, releasing glucose, making it available for the body to use. During meals and a great influx of nutrients, we secrete insulin to store excess food in various places in the body. In between meals, insulin's counterpart, the hormone glucagon, promotes the hydrolysis of stored fat into fatty acids, which are released into the bloodstream. Those are transported to all parts of the body. Cells that need energy take up the fatty acids. Fatty acids are moved to the mitochondria, where they undergo beta oxidation, forming acetyl-CoA. Acetyl-CoA then goes into the Krebs cycle and is converted to CO2, producing NADH, which is then made into ATP. The video Sugar the Bitter Truth reminded us that we are in an epidemic, in the midst of an epidemic of diabetes type 2 with 10% of the adult population affected. Dr. Lustig tried to make the point that it's not fat which is the major culprit in our diet. He's showing this slide that calories from fat as a percentage has decreased over the last 30 or 40 years, whereas obesity has massively increased. Instead, he points out that the real culprit molecule is fructose, which is part of sucrose. Sucrose is a disaccharide made up of one molecule of glucose and one molecule of fructose, and also high fructose corn syrup provides a lot of fructose in our diet, and that it's really the portions that have grown so supersized and the food industry's ability to hide sugar in processed foods that make us want to eat them and is causing a lot of this damage. Glycolysis in the citric acid cycle is often referred to as central carbon metabolism. Here you can see glucose, and here's the citric acid cycle. And in this overall metabolic map of all the different types of metabolism that cells undergo, it's always centered in the middle, and it's part because the branches of the intermediates, the things that are tried to made, whether you're trying to make nucleotides, amino acids, or lipids, all branch off from this central metabolic pathway. On this slide we see an overview of the molecular details of glycolysis. We start with one molecule of glucose with 6 carbons. We will finish with 2 molecules of pyruvate, each with 3 carbons each. We're going to invest 2 molecules of ATP, producing this molecule fructose 1,6 bisphosphate. And then that is going to be cleaved. In later steps, we're going to have steps that produce a molecule of NADH and 2 steps of ATP for each of the 3 carbons, so double that. We're going to put in 2 molecules of ATP. We'll get out 4 molecules of ATP for a net of +2. This NADH in the presence of oxygen will be worth more ATP. In the absence of oxygen, it's important to replenish the NAD+, and bacteria have done this by fermentation, and our muscles convert pyruvate to lactic acid in order to regenerate NAD+. There's no oxygen at all involved in these steps of glycolysis. The production of ATP in these steps is referred to as substrate level phosphorylation, with specific enzymes attaching inorganic phosphate to a molecule of ADP. A different enzyme is used in step 7 versus step 10. And substrate level phosphorylation stands in contrast to oxidative phosphorylation, in which all of the molecules of ATP are created via a proton gradient being converted to ATP by the enzyme ATP synthase. The last points to make is this first molecule of ATP traps glucose inside a cell. Via transporters, glucose can move in and out. If glucose is phosphorylated, that means it's trapped inside the cell and will be used by that cell. However, by step 2, it can still go into various metabolic processes. However, when we invest that second molecule of ATP, then it's committed to going through glycolysis down to pyruvate. This, in part, that's because now you've invested a lot of energy into the production of this molecule, and you have to recover some of that. And secondly, just because this step is so energetically downhill that it's hard to go in the reverse direction at any appreciable rate. In the next slide, we'll follow the fate of the pyruvate molecules. So sugars get converted to pyruvate in the cytosol of the cell. Pyruvate gets transported into the mitochondria, and there it is converted to acetyl-CoA, which then enters the Krebs cycle. Fatty acids are picked up from the bloodstream. They bypass the cytosol and are transported directly into the mitochondria. Here they undergo beta oxidation, cleaving 2 carbons of the fatty acid off at a time, producing multiple molecules of acetyl-CoA. Once the acetyl-CoA molecules are made, whether from sugar or from fat, they are processed in exactly the same manner. Here we see the molecular overview of the citric acid cycle. We have acetyl-CoA, 2 carbons, joining with oxaloacetate, a 4-carbon molecule, into the 6-carbon sugar citric acid. This 6-carbon sugar will be converted back to the same 4-carbon sugar oxaloacetate by a series of oxidation reactions. During some of those, carbon dioxide will be evolved. These represent the complete oxidation of the sugar molecules down to inorganic carbon. On other oxidative steps, high-energy electrons are formed in the stage of NADH or FADH2, which is not quite as energetic as NADH. Each of these molecules of NADH will be responsible for about 3 ATP molecules, and FADH approximately 2 ATP molecules. We also do substrate-level phosphorylation. We form a molecule of GTP from a molecule of GDP, and in some cases, GTP can be used to power reactions like ATP. On this slide, we see the beta oxidation cycle in which fatty acids, long fatty acids, are joined with a molecule of CoA to form a fatty acyl-CoA. It's going to enter into this cycle, which is 4 steps, and in each step, 1 molecule of acetyl-CoA will be produced, and the resulting fatty acid CoA will be 2 carbons shorter. This process is highlighted. The 2 carbons here are red. These are the 2 that get converted to acetyl-CoA, which can then enter the Krebs cycle. Notice that in this process we're also producing molecules of FADH2 and NADH, which will be used to make ATP, just like the NADH that will result from the oxidation of acetyl-CoA. This slide is highlighting the fate of the NADH that has been produced. Remember, we're taking acetyl-CoA, burning it to carbon dioxide, producing NADH, which has high energy electrons. These electrons will be donated to an electron transport chain, which will be used to establish a proton gradient, and that proton gradient will be used to synthesize ATP. These high energy electrons will have their energy degraded during the electron transfer reactions. Eventually, these very low energy electrons will be deposited on molecular oxygen, the reason we breathe. It's going to produce water as a byproduct. And in this slide, we just highlight the importance of NAD+ for anaerobic metabolism. Without oxygen, your NADH, which is normally a valuable molecule, is not valuable at all. And in fact, we need to get rid of it so that we can continue to produce glycolysis and get at least the 2 ATP for each molecule of glucose. That's net. So what happens to these extra electrons on NADH? Where do we dump them in order to regenerate NAD+? Well, the cells take pyruvate, which is the breakdown product of glycolysis, dumps the electrons on the pyruvate, making lactic acid, the thing that gives you cramps in your muscles when you're exercising. Now you've got NAD+ that can work in the reaction, and you can keep generating the small amounts of ATP that is possible anaerobically. Okay, in the second video we talked about the details of oxidative phosphorylation. Again, we have high energy electrons sitting on molecules of NADH. They get donated to this electron transport chain, which are 3 major protein complexes that exist in the inner mitochondrial membrane. As the electrons go onto these protein complexes, they undergo a conformational change which picks up protons from the inner mitochondrial matrix, pumps them to the outside, the inner membrane space. So you have a low pH here. That means a high concentration of protons. Eventually these protons will flow back through the ATP synthase to synthesize ATP from ADP. Again, there are 3 protein complexes that are each pumping protons. Now that when it gets done with cytochrome C oxidase, the electrons have low energy. They're deposited on molecular oxygen as a final electron resting place. On this next slide, which looks just the same, we highlight the fact that this is called coupled vectorial processes. We have the electron transport in the membrane, pumping protons across the inner mitochondrial membrane, having the protons diffuse back down their electrochemical gradient, driving ATP synthesis. In this process, these combined processes are referred to as coupled vectorial processes. This slide shows some of the details on the electron shuttling between the electron proton pumping complexes. First one being NADH dehydrogenase complex, and the last one being cytochrome C oxidase. The first electron shuttle is by a small molecule ubiquinone, which is hydrophobic, and it resides here in the hydrophobic interior of the lipid bilayer. The molecule is able to diffuse freely back and forward, but since it's hydrophobic, it cannot escape into the aqueous environment. When it collides with the cytochrome BC1 complex, electron will be donated here. Cytochrome C is a protein that is attached to the membrane by a lipid tail. This lipid tail is stuck in the lipid bilayer, so I'll call this the balloon on a string model. In cytochrome C, the protein is free to diffuse back and forth along the surface of the membrane, but it cannot leave because that would expose this lipid tail again to the aqueous environment. Neither ubiquinone or cytochrome C pump any protons. And it's here at cytochrome oxidase where if cyanide poison binds to the spot where oxygen normally would, it prevents oxygen from binding. That means the electrons don't get deposited on the oxygen. They'll start to back up in the system. You'll no longer do the Krebs cycle. You'll no longer do any electron transport, and you will run out of ATP very quickly. This slide is highlighting the relative energies of the electron on the various protein complexes. These protein complexes have different redox potentials or electron affinities. These high energy electrons are first deposited on the NADH dehydrogenase complex, and the energy is degraded by binding to proteins of lower electron affinity until eventually they get deposited on oxygen. Again, the inputs to respiration are the oxygen that we breathe and the food that we eat. The outputs, just as if we're burning organic material, is carbon dioxide and water. Now we've created this proton gradient by the proton pumping, and we're going to use it to drive the synthesis of ATP. This slide talks about the two components, two driving forces which are both in the same direction, which reinforce to make the proton diffusion energetically downhill with sufficient power to synthesize ATP. The first part is the electrical part. If you have a lot of protons out here, you have a lot excess of positive charges relative to the other side of the membrane, and so that removing a positive charge from here, combining it with a negative charge here, is energetically favorable. It's also minimizing repulsion on this side. And then here's the chemical portion of the gradient, a high concentration of protons, a lower concentration of protons. So that is part of, again, the electrochemical gradient. The Nernst equation describes the relative importance of these two, but we did not explore that equation in class. Here we see a depiction of the three-dimensional structure of the ATP synthase molecule, or also known as F1F0 ATPase. Here is the inner mitochondrial membrane. These are the two lipid bilayers. The protons from the inner membrane space flow into here. That causes a rotary motion of this red stalk. The green part does not rotate, but the rotor inside here moving causes conformational changes in the alpha and beta subunits, and these conformational changes will cause ADP to bind, ADP and inorganic phosphate to be combined into ATP, and then ATP being released. The total oxidation of a glucose molecule produces between 30 and 38 ATP, with sort of the most commonly cited number 36. Some of the things to remember: that in the absence of oxygen, the net yield is very small. And we discussed some of the reasons for the disagreement between different accounting schemes. that there is no one number that's valid at all concentrations of glucose, NADH, cyclic AMP, protons, specifically talking about two successive protons diffusing down the electrochemical gradient, and how there's a different driving force for the two of them because the electricity has changed, the electrical forces have changed, and the chemical gradient has changed. We highlighted the similarities and also talked about some of the differences of photosynthesis with oxidative phosphorylation. Photosynthesis, the inputs are light, water, and carbon dioxide. The outputs are oxygen and biomaterials, and for the plant, ATP to power its metabolic processes. Photosynthesis takes place in two stages. One of the light reactions, this is the photo-initiated charge separation reactions, and then the dark reactions, which can happen at any point in the day, where the ATP and the NADPH produced by the light reactions are used to incorporate inorganic carbon dioxide into organic materials in a process referred to as carbon fixation. On this slide, we see the protein complex, the reaction center, which is the site of the initial photo-initiated energy-producing step. That energy-producing step is an electron transfer between the special pair of chlorophyll molecules. There's the special pair, which is going to transfer an electron down to quinoline molecules, leaving a positive charge here and a negative charge here. Light-driven charge separation. This is raising the potential energy of the system. As we know, these opposite charges attract and want to recombine, but we're driving them further and further away. This electron hole, this positive charge, is replaced by electrons ultimately from water. So that would make a neutral molecule. Another photon would come in, another electron transfer reaction would occur. Then you're going to have two electrons in a quinone molecule. The quinone can diffuse out into the membrane and transfer the electrons to a proton pumping protein complex that is inside the membrane here. We also talked about the surrounding proteins called the antenna proteins, which have 200 to 400 molecules of chlorophyll, and this is to increase the absorption cross-section for each photon. When a photon hits, it will hop around without transferring electrons, but the photon hops from these chlorophyll molecules, eventually making its way to the special pair. The energy levels of these chlorophyll molecules are finely tuned such that it's essentially an energetic funnel with forcing the photon to eventually go to the lowest energy position, which is the special pair. The energy tuning is performed by the immediate amino acid environments of the antenna protein. Things get a little more complicated in photosynthesis because there's two photosystems. We've just been talking about photosystem 2, which is where things start off. There's also a photosystem 1, which absorbs another photon that's going to do some more of the biochemistry. Here, let's just remember that here we have produced a positive and negative charge. The negative charge has gone on to quinone. It goes on to this protein complex, which is going to pump protons. These protons will later be used to synthesize ATP. Instead of cytochrome C, which is tethered to the membrane in photosynthesis, there's a protein very similar called plastocyanin that's going to accept the electron and transfer it to photosystem 1. Photosystem 1 will then absorb an electron, transfer to ferredoxin, and these high energy electrons will not produce ATP. They'll be used to synthesize NADPH from the molecule NADP+. This molecule stores electrons just like NADH does, but the presence of this phosphate group here marks these electrons for biosynthetic uses as opposed to donating it to the Krebs cycle to make ATP. The requirement for two photosystems and two photons will reflect, and we'll see this in the future slide, is the fact that we're grabbing electrons from water. We're going to split water open, grab the electrons, produce oxygen as a byproduct, and these are very low energy electrons. This molecule is very high energy electrons. To bridge the energetic difference between these two, we need two photons. These are also coupled vectorial processes. We have electron transport, proton pumping, protein diffusion down the gradient to synthesize ATP. And this is the Z scheme. Again, you can see that the electrons on water are at a very low energy, and it's quite an engineering feat to be able to use low energy electrons such are present in water for these processes. A single photon gets you up to about minus 600 millivolts in terms of redox potential, which is not enough to synthesize NADPH. So as the electron energy is being used to pump protons, and through various electron acceptors of lower affinity, it's going to use about half its energy here for ATP synthesis, and then it's going to be promoted to another excited state by the second photon, and now it's at a high enough level for this biochemistry. The energy of the individual photons is the same, somewhere in the near infrared, between 650 to 700 nanometers is the minimum wavelength required for this process. So normally these two photons are used to make ATP and NADPH, but sometimes the cell doesn't need as much NADPH, so we're going to talk about a short circuit here where even the electrons from photosystem 1 are used to synthesize ATP. And here you can see the cycle. This is a little less efficient, but nevertheless it's more useful for the cell to produce ATP if that's all that's needed. So here photosystem 1 is using ferredoxin not to go ahead and produce NADPH, but transfers the electron to the plastoquinone, which then resumes the electron transport process in the inner thylakoid membrane of the chloroplast. Here we see the molecular overview of the carbon fixation reaction. Again, you're not responsible for particular structures, but we have carbon dioxide, 1 carbon, and a 5-carbon sugar, Ribulose 1,5-bisphosphate. It's going to be joined together. It's going to undergo a rearrangement, and we'll get 2 3-carbon molecules of sugar called 3-phosphoglycerate. All this biochemistry occurs as part of a rather complicated Calvin cycle, and for the numbers to work out, many cycles need to be achieved. The main thing we want to do is just check that the numbers work out or roughly correctly. We have 3 molecules of carbon dioxide going through the cycle 3 times, producing 1 3-carbon molecule of glyceraldehyde 3-phosphate in total. And to do that, we're using quite a lot of ATP, 9 molecules of ATP, 6 molecules of NADPH, and this is a considerable investment but it takes this very oxidized carbon and produces a very reduced form, electron energy rich molecule. These next 2 slides are going to highlight the similarities and the differences between photosynthesis and respiration or oxidative phosphorylation. In both cases, we have high energy electrons. One is from NADH, one is from the absorption of a photon. We're going to capture some of that energy in the form of a proton gradient, which is then used to synthesize ADP. The source of the high energy electron is the metabolism of foodstuffs in mitochondria, or the absorption again of the photon in photosynthesis. But in other ways, it's almost the opposite process. Here plants are going to consume water and produce oxygen, whereas we consume oxygen and produce water. It's going to use carbon dioxide to make sugars. We're going to take sugars and produce carbon dioxide as a byproduct. And it's probably just worth pointing out that by removing carbon dioxide from the environment, we will be minimizing whatever effects it might have on global warming, and many plans for dealing with excess carbon dioxide in the atmosphere do revolve around using engineered plants to trap the carbon dioxide in the environment into non-volatile molecules, which may have other uses as a side bonus. I leave you with this slide on this section just as an overview of everything that's happened. I won't go through it, but many students in the past have said this is a very useful slide for them to think about and check their understanding of the various processes involved with glycolysis, the citric acid cycle, and oxidative phosphorylation. In the next lecture, we talked about membranes. We talked about how they're primarily lipid bilayers with hydrophilic heads on the outside, hydrophobic tails on the inside, that there are two leaflets. Hydrophobic ends from both are near each other, creating a hydrophobic environment, and that proteins can be as much as 40% of the mass of a membrane, and we talked about the functions that the various proteins had. Highlighting on this slide is the continuity between the fluid outside the cell and in various compartments inside a cell. Also here we've colored the two leaflets of the lipid bilayer different colors, black for the exoplasmic phase and red for the cytosolic phase. And as we see, the endoplasmic reticulum, that the relationship of the membranes is maintained, and when we see a vesicle budding and then it's going to fuse, the red will always be facing, whether it's here in one of these organelles or in the membrane, the red side is always facing the cytosol. The black leaflet is pointing outwards or inwards to the interior of these various organelles. It's also highlighting the fact that mitochondria, the nucleus, and chloroplasts not pictured have double membranes, while as the rest of the organelles are enclosed by a single membrane. This was a proposed mechanism for the generation of a double membrane nucleus where DNA was affixed in a prokaryote to the surface invaginated and enclosed it. But I point this out only mostly as a memory aid, not as a mechanism that we have to study. For mitochondria, the proposed mechanism is that it's a bacteria that got engulfed by phagocytosis and then eventually lost its independent character and became part of the cell. Mitochondria have their own little genomes and the ability to do transcription and translation that gives further evidence to, or further support for the notion that current mitochondria represent evolutionary vestiges of bacteria that were engulfed by eukaryotic cells. One of the main functions of a membrane is to create distinct environments. There's a clear difference between the inside and the outside in terms of molecules that are present. And with organelles, you can even have multiple different environments inside a cell, allowing different chemistries to occur in different compartments. Membranes will not only maintain these differences, they're also partially responsible for their creation. Three major membrane lipids that we talked about are phospholipids, cholesterol or other steroids, and glycolipids. Fatty acid chains, in this case, being attached to a molecule of glycerol with a polar head group, a phosphate group, and in this case choline, a molecule that's like an amino acid. We talked about the kinked hydrocarbon chains, preventing close packing of these phospholipids. So if you have more lipids with unsaturated fatty acid chains, the membranes will be more fluid. Cholesterol has a very small hydrophilic group, but this and the rest of the molecule is very hydrophobic. It's shorter than these, and we'll see the packing of cholesterol with fatty acids on the next slide. Glycolipids have still a three-carbon backbone. They have hydrocarbon tails attached to it and sugars attached as polar head groups, which are particularly abundant in the nervous system. Here we see how cholesterol can help fill spaces between two adjacent phospholipids that have unsaturated hydrocarbons. This helps maintain the relative impermeability of membranes, preventing unwanted items from diffusing across a lipid bilayer. In aqueous solutions, lipid bilayers spontaneously form spheres which have this is a cross-section through there, a lipid bilayer with waters on both sides, and it closes up on itself so that there's no exposed surfaces here of the hydrophobic regions to the aqueous solution. For the lipids in the membrane, there is a great deal of lateral diffusion, also the proteins there, but there's essentially zero flip-flopping. So the two different leaflets maintain different pools of phospholipids in their boundary, and one way of depicting this lateral diffusion is this experiment which shows the evolution in time of a mixture of lipids, one of which is labeled with fluorescence groups. These lateral interactions between head groups cause certain lipids to aggregate with each other, and we have this soccer ball appearance that's present in the second fluorescence micrograph. One of the main ways of studying the diffusion rates quantitatively is to have cells in which there are fluorescent groups attached to the various lipids, or it can be the proteins that are diffused in the lipids, to take a laser, focus it down to a very small spot, maybe a micron in dimension, blast this area of the membrane with photons, so many photons that it will cause a destruction, a photodestruction of the fluorescent molecules. So without any fluorescence there, you have a region of that's white in this picture, but in actuality not fluorescent green. Then over time, these photodestroyed lipids will diffuse into this region, and the non-photobleached lipids will diffuse into that area, so that you can look at the initial photobleach and you can time the recovery, and that from that you can back calculate diffusion constants for the lipids or the particles of interest inside a membrane. This method is referred to as FRAP, which stands for fluorescence recovery after photobleaching. We next went on to a discussion of the proteins in the membranes. We talked about their structures first and then some of their functions. Many membrane proteins pass the membrane with a single alpha helix. There are classes of proteins like the G protein coupled receptors that have 7 alpha helices. Here's a beta barrel channel which produces a large pore in a cell. There are a relatively very small number of proteins that exist in one half of the leaflet as opposed to both. These are mostly, that I've seen, regulators of the curvature of the cell membrane. We've seen examples like cytochrome C of proteins being attached through single lipids to the lipid bilayer so that it can diffuse laterally but not escape out into the aqueous environment. There are other kinds of lipids that can be used. This is called a GPI linkage, which has sugars and lipids. And then there are also proteins which attach to proteins that are themselves inside the cell. We talked about various types of functions that the membrane proteins do. There are transporters and channels for moving molecules in and out of cells. There are anchors which interact with both the extracellular matrix and the inner cytoskeletal scaffolding of the cell to provide rigidity and structural strength to the cell. There are receptors which bind ligands and then transmit signals to the inside of the cell as a means of communication with the outside world. Sorry about that. Proteins are anchored in membranes by having runs of hydrophobic amino acids. Here, about 15 to 20 hydrophobic amino acids will position this protein in the membrane. There are probably hydrophilic amino acids here, and if this peptide is to move either up or down a little bit, or this portion of a protein, that would cause unfavorable interactions. Well, let's say if it's moving down, these hydrophobic amino acids would then interact with either the solvent or these hydrophilic head groups, and so that the energetic minimum is a protein positioned in this way inside the membrane. All alpha helices don't need to be completely hydrophobic. They just need to be hydrophobic on the side next to the membranes. Here we have amphipathic alpha helices, which have hydrophobic exteriors, but colored in red here are hydrophilic interiors. And now we have a pore that's going to allow the passage of a hydrophilic molecule across the membrane. We talked about molecules and their ability to cross pure lipid bilayers. Small hydrophobic molecules pass as almost if it's not present. Small and uncharged polar molecules, water, glycerol, and ethanol are examples. Those go through at a reasonable rate as well. Larger molecules like amino acids, sugars, and nucleosides generally do not pass through the lipid bilayer, but need to be transported by a protein to enter into the cell. And then there's the charged molecules. In this case, they're mostly looking at ions, and these are extremely resistant to passage across a pure lipid bilayer. Now, the way that proteins assist molecules like glucose and other things to cross the membrane is a transporter protein, where you have a solute binding site, a molecule can bind, there can be a conformational change, and then a molecule can be released on the other side of the membrane. Channels are going to mostly allow small molecules to pass. Channels are either open or closed. Once they're open, these molecules will always flow down their electrochemical gradient. Slides highlighting passive transport, in which molecules are moving down their concentration gradient. Here you can see many blue molecules, for instance, and only a single one on this side. And so this is even though it's using a transporter protein to do passive diffusion, it is going down its concentration gradient. If you're going to go against your concentration gradient, you need the input of energy. And again, that's called active transport. This slide is showing an example of passive transport, where the binding of the solute molecules causes the conformational change and release on the other side, again going down the concentration gradient. If you're going to go against your concentration gradient, here we're going to try to transport this yellow molecule over to this side where it's already more present. You can do that by paying coupled transport. You're going to transport some molecules down their electrochemical gradient, which can power the pumping of a molecule against its concentration gradient. Of course, you can utilize ATP hydrolysis to pump molecules. This is the mechanism for one of the primary users of ATP in the cell, the sodium-potassium exchange pump, and there are light-driven pumps, where the absorption of a photon will cause the isomerization of a molecule, which will lead to a protein structural change, which can lead to the transport of a proton from one side of the membrane to another. Showing a little bit more details on the sodium-potassium pump. Sodium is prevalent on the outside of the cell. Potassium is present on the inside of the cell. Once the things flow through channels, this gradient needs to be replenished. The way that's done is by the hydrolysis of ATP, pumping out sodium despite its great abundance outside, and pumping potassium inwards to reestablish the concentration gradients that are present in the normal resting equilibrium state of cell membranes. Important example of how glucose is absorbed by the body. In the digestive tract, the glucose is at a rather low concentration. But the epithelial cells lining the digestive tract want to grab all the glucose from the digestive tract. So this glucose is initially being transported up its concentration gradient. We pay for that energy that's required to move glucose into the cell by allowing the very plentiful sodium that's present in the gut lumen to go down its concentration gradient, and that's how we pay for moving glucose into the cell. Now, to share this molecule of glucose with the rest of the body, it's allowed to transport passively through a transporter out into the extracellular fluid. Here it'll be picked up by the bloodstream and transported to other parts of the cell. The sodium needs to get out of the cell, otherwise you're going to get too large a concentration of sodium inside the cell, and here we're paying with ATP to reestablish the sodium and potassium gradient. We concluded this section with some talk on ion channels and how they're regulated, voltage-gated channels, where changes in voltage around the membrane can cause the channel to open or close, ligand-gated systems where a ligand binds a receptor which causes a conformational change and opens up the channel, and also mechanically gated channels such as in the ear cells or pressure or pain sensitivity receptors in your fingertips and other places. We talked a fair bit about the potassium channel, which has been solved at high resolution X-ray crystal structure. We talked about how this protein has solved some of the thermodynamic problems associated with producing a channel that's selective for potassium. One was the stabilization of positive charges in the middle of this hydrophobic membrane. Part of the reason the way it's done there is by focusing electric fields, negative electric fields, using the helix dipole into the vestibule region, stabilizing positive charges. And then the selectivity filter, which allows the large potassium to go through but prevents the smaller sodium to pass through. And that's explained thermodynamically. Again, there's 3 stages of carbonyl binding that constitutes the selectivity filter. First, we're going to look at the situation for sodium and potassium in the vestibule. Here there's a lot of water around it, and these ions are hydrated by at least 4 molecules. When you get into the selectivity filter, potassium is big enough to be hydrated or coordinated by these carbonyl groups that are part of the protein amino acids lining the channel. Sodium is too small. It's only going to be interacting with 2 carbonyl groups, making this a much more energetically favorable position, and so it's less favorable that if a sodium ion happened to jump into this first position, that it wouldn't proceed to the second and third position, but would be more likely to return to the vestibule. In axons, the action potential, which travels down the length of the neuron, is achieved by the successive opening of ion channels, primarily sodium initially, which is going to cause a particular ion channel will open. That will create an electric field, which will cause nearby channels to open. That's going to allow new ions to rush, sodium ions to rush in. That's going to, again, create an electric field change. That's going to cause more channels to open, and so on. Finally, as the action potential comes down here to the end of the nerve, the Proton, and it's going to communicate with a postsynaptic cell, the action potential will cause a voltage-gated calcium channel to open. Calcium will flow in. This will cause a change in conformation of some of the proteins in the vesicles which hold the neurotransmitters. That will spur fusing with the plasma membrane and release of the contents of the vesicle into the synaptic cleft. These ligands, these neurotransmitters, can then bind to these ion channels and allow ions to flow in and out, creating the condition for the start of some biological event. It could be another action potential. It could be a muscular contraction. It could be a whole host of things. And in class, we talked about 2 diseases which result from inappropriate ion channel function. In this case, cholera, which is a toxin from the bacteria Vibrio cholerae. It infects your epithelial cells, and it leads to inappropriate expression of the chloride channel, which is known as the cystic fibrosis transmembrane receptor. When this channel is placed here in the membrane because of the cholera toxin, now it's pumping chloride out. To an attempt to maintain electroneutrality, sodium is going to go out. This is decreasing the osmotic concentrations inside the cell, decreasing it inside the cell, increasing it out here in the digestive lumen. So water will flow, and it's this flow of water which causes diarrhea and the dehydration of the cells in the environment, and then from the bloodstream and your whole body. Cystic fibrosis, on the other hand, is the opposite. Here you have, normally, the cell wants to pump chloride ions out so that sodium ions will follow and water will follow, and that will keep mucus relatively hydrated and allow it easier to clear from your lungs as a protective coating. When the chloride channel cannot function, chloride's not going out, sodium's not going out, water's not going out, the mucus is going to get dehydrated. It's going to become thick and sticky, and it will become a breeding ground for bacteria in the lungs of cystic fibrosis patients. The next video talked about different organelles and how proteins get sorted to the various organelles. We spent a few minutes talking about different organelles, and you would want to be familiar with the function of all these organelles. There are 3 basic different mechanisms that proteins are transported. They can be transported intact through large openings like going into the nucleus. They can be unfolded and transported into organelles like the mitochondria or the chloroplast. They can be synthesized directly into the ER and then have vesicles budded to meet up with other organelles and the plasma membrane. A difference between these mechanisms 2 and 3, here the protein is fully synthesized in the cytosol, unfolded and transported. Here it's synthesized directly into the endoplasmic reticulum. Proteins know where to go by containing little localization signals, which are generic in their function, and if you use the tools of molecular biology to swap them, the location of the proteins will change. Even if they're not functional in this environment, you're not responsible for any of these sequences that are localization signals for the nucleus or the lumen of the endoplasmic reticulum or the mitochondria, but these are seen over and over again in many proteins, along with slight variations. Sometimes the signal sequences can be maintained with the protein. Most of the time it's cleaved off for a functioning protein. Here we see a large picture of the nuclear pores. Again, this distance here across a pore is enough for about 50 ribosomes, so this is not at all to scale here. The way that proteins are imported specifically through this pore is that they contain the localization signal. A transport receptor can bind to that localization signal. It can interact with these tendrils emanating from the nuclear pore, and it can help transport the protein to the inside of the nucleus. Here, ATP or GTP is hydrolyzed, and that will pay the energetic cost of transporting the receptor back outside into the cytosol. Now, because it's so large, these many molecules are capable of going in and out of the nuclear pore on its own, but that would be down diffusion gradients and stochastically. Here, this is the very specific import of proteins into the nucleus. In mitochondria, proteins are synthesized and unfolded with the help of these chaperone proteins and the hydrolysis of ATP. The localization signal binds to a receptor in the mitochondrial matrix, which then translocates or diffuses in the membrane to this place of close contact between the two, the outer mitochondrial membrane and the inner mitochondrial membrane, and proteins can be threaded through, pulled, if you will, again by this chaperone protein and the hydrolysis of ATP. Once inside the mitochondrial matrix, the localization signal is cleaved and the protein will fold into an active form. For proteins going to the inner mitochondrial membrane or the inner membrane space, in addition to this localization signal, they have a second series of amino acids, this orange sequence, which is hydrophobic, and that indicates its position should be in the mitochondrial membrane. And then you can, and here you have produced a protein that is present in the mitochondrial membrane, or this sequence can be cleaved off, releasing a protein to be free in the inner membrane space. For the endoplasmic reticulum, a protein is started to be synthesized out in the cytosol, and as this localization signal for the endoplasmic reticulum emerges from the ribosome, it's bound by this protein called the signal recognition particle. This arrests amino acid synthesis, protein synthesis, until that this complex now can bind to one of the ER translocation channels, and then synthesis will continue, threading the protein into the lumen of the endoplasmic reticulum. The presence of a hydrophobic stop transfer sequence would still allow the protein to be synthesized, but it would now be maintained in the membrane of the endoplasmic reticulum, as it would be maintained as vesicle budding and then in fusion with the plasma membrane if it's going to be a surface protein. The part of the protein that is in the cytosol right now will always be in the cytosol. So if this fuses with the plasma membrane, this part of the protein will be facing the outside world, and this will be facing the inside of the cell. Once a protein is in the endoplasmic reticulum, it can stay there, or it can be sorted to other locations like the Golgi via vesicle budding and fusion. If it matures through the Golgi apparatus, then it can be part of vesicles which might be bound for the surface of the cell, confused with endosomes to process materials that have been endocytosed, or it can end up also in other organelles. The movements of the vesicles themselves are mediated by proteins which coat the vesicles. COP2 drives, here's the nucleus over here, here's the outside world. COP2 is the protein which coats vesicles if it's going to move away from the nucleus. COP1 is the protein that's going to take vesicles and move it towards the nucleus. These interact with various motor proteins, either kinesins or dyneins down here, which are going to walk along microtubules in an energy-dependent process. And this overall produces the locomotory force which drives vesicle movement. This slide shows the microtubule network, which are often referred to as the highways of the cell. You have a nucleus maybe somewhere around here. This is the centrosome. This is the organizing center of microtubules. Note that there are plus ends and minus ends. Microtubules grow on the plus ends, and these various molecules will move one way or the other, depending on the motor protein that's interacting with the microtubules. Vesicles move from one place to a second by a process of budding and pinching off the membrane, coming in close contact with the membrane, fusing and transferring molecules that are free in the lumen, dumping them out into the lumen of the new organelle. Proteins that are present in the membrane will also be present in the membrane, and in the same orientation is maintained. Moving materials in and out of the cell can be achieved by exocytosis for outward or endocytosis for inward, and these are just variations on the vesicle fusion and pinching off mechanisms. The way that a vesicle knows where to fuse is mediated by these snare proteins. There are sort of donor and acceptor snare proteins that have to interact with each other. Once they start their interaction, they start to have greater interactions that pulls the vesicle in close contact with the membrane, and then it's energetically, thermodynamically favorable to have a fusion of this vesicle, and then releasing the curvature, the strain energy of this membrane will flatten it out over time. The specificity of where to dock is, again, mediated by these snares. This cartoon has different shapes on the snares, a triangle versus a circle. The circle one's going to dock with this target snare. The triangular one's going to dock with this target snare. But that would prevent, for instance, this vesicle from fusing with compartment A. This slide has a very detailed look of how proteins are sorted for the plasma membrane. I'll go through it very quickly, but you should look at all the detail that's written out here. Proteins synthesized in the endoplasmic reticulum, transported by vesicles through the Golgi apparatus, then finally released as vesicles. They can secrete, they can fuse with the membrane immediately upon contact, or they can be held in a regulated way until a special event occurs. The process of endocytosis is the pinching off of a membrane, taking up of materials from the outside world, fusing with other transport vesicles to create a late endosome, then a lysosome, in which materials are degraded into useful molecules for the cell. The process of endocytosis is mediated by a protein called clathrin, which is going to, we'll see, have a geometry that forces the invagination of part of the plasma membrane. In this case, a receptor, which is bound its molecule, is present in this area. It's going to get trapped on the inside of this vesicle. It's going to go into the endosomes, where a pH shift will cause dissociation of the bounty from the receptor. The receptor will be recycled to the plasma membrane for importation of more molecules. The cycle of the receptors can happen many hundreds to thousands of times per protein. Now this molecule is going to go and be transported to a lysosome, where nucleases, proteases, and lipases are going to chew the material up again into useful molecules. Here we see the geometric constraints of clathrin and how the close packing of clathrin and receptor molecules forces the membrane to invaginate in strictly to allow close packing of the clathrin molecule, which is very wide out here. Eventually the stalk region is going to get pinched off in an ATP, requiring process by a protein called dynamin in a boa constrictor type move. Once the endocytic vesicle has made it into the cytosol, the clathrin will be released and you have what's referred to as a naked transport vesicle. Another mechanism for importing things into a cell, in this case an immune cell, is going to engulf a bacteria via the process of phagocytosis. The actin network is going to build itself and push out the membrane to completely surround the bacteria, and now this bacteria is ensconced in a double membrane type structure. It will fuse with other vesicles to produce a number of molecules which will kill the bacterium, and it will be cleared from the system. Neutrophils are examples of the phagocytic white blood cells that are involved with the clearing of bacteria. We concluded this section by talking a little bit about lysosome, which is a organelle which has lots of nasty proteins which are active in the acidic environment inside lysosomes, but if the lysosome was to burst and the enzymes released in the cytosol, they would be inactive at the more alkaline pH. This acidic environment is caused by running an ATP synthase in reverse, essentially. Here we're going to hydrolyze ATP to pump protons against its concentration gradient. Remember, in oxidative phosphorylation, protons went down the concentration gradient to synthesize ATP. This is an example of reversibility. In the next section, we talked about cell signaling, a process also called signal transduction, or as the book calls it, cellular communication. We have here a generic signaling pathway which consists of a ligand which binds a receptor, which transmits a signal via intermediate signaling proteins, most often by phosphorylation of 3 amino acid residues, serine, tyrosine, and threonine, all which have hydroxyl groups, and eventually a kinase here is going to affect an effector protein. Maybe it's a transcription factor, which will lead to altered gene expression. It can affect a metabolic enzyme, converting an active protein to an inactive, or vice versa, leading to altered metabolism, or proteins themselves can lead to, for instance, altered shapes. There are different types of signaling. Sort of the classic one that we've just talked about is endocrine signaling, where a hormone molecule is released from a distant part of the body, is transported via the bloodstream to all parts of the body, and then the hormone or ligand binds to a receptor of a target cell, and some physiological event will occur. In contrast, the shortest range interaction are ones where the ligand is not secreted out into the extracellular matrix or extracellular fluid, but rather is bound to another cell, and so these two cells are signaling each other via a ligand molecule and a receptor. Paracrine signaling releases molecules into the extracellular fluid, but it's only transported locally. It's not picked up by the bloodstream, and so these typically have ranges of at most a millimeter, probably less. Nerves are a specific example of a signal that can go over a long distance overall because of the action potential, although the chemical synapse is always a small region. We talked about the specificity of signaling in a number of ways. This slide was showing that the same molecule, acetylcholine, can bind to different molecules and produce different results: contraction, secretion or heart rate. And that's in part because these signaling pathways are modular and can be hooked up to different effector proteins in the last stages of the signaling pathway. We also spoke about the kinetics of a signaling process, contrasting a fast response in which a signaling event merely alters the function of an existing protein versus a more typical and slower response where the ligand will eventually activate a transcription factor which binds DNA, which will cause RNA to be synthesized, which will cause protein to be synthesized, and then eventually you'll have an altered phenotype. Slow minutes to hours to 24 hours is a very reasonable time for something to occur via this pathway, whereas here, just changing the function of a protein, this can happen in as little as a few seconds. Most signaling ligands are hydrophilic and bind to receptors that are present on the surface of cells. There is a class of hormone receptors, in particular, like estrogen, like cortisol, that are proteins that exist either in the cytosol or the nucleus. The molecule, which is the signaling molecule, like estrogen, is hydrophobic. It can pass through the membrane, it can bind through the protein, and the protein can take actions almost immediately. This slide highlights that the primary way that information is transferred within a cell is by the creation or the removal of phosphate groups. And the enzymes which put phosphate groups in proteins are called kinases, and although the signs can change, most people think of a kinase, a phosphorylated protein, being active, being turned on. Now it's got some activity. Eventually, the kinase's counterpart, a phosphatase, will remove the phosphate group from the protein, causing a conformational change, causing a change in activity. The source of these phosphoryl groups is ATP, which is cleaved into ADP, and a phosphoryl group which can be transferred to a target protein. There's another set of proteins which are regulated in a very similar way, but rather than having just a part of the phosphoryl group of ATP being covalently attached, it binds non-covalently a molecule called GTP, which can then be hydrolyzed into GDP. The net difference between these two states is one phosphate group, just like it is over here, and this is another very frequently used mechanism for transmitting signals from the outside world to the interior of a cell. While the estrogen receptor that we saw had the advantage of being very simple, the complexity of having a lot of proteins involved in a signaling pathway allows for a lot of modulation of a signal of the binding of an external ligand. You can do things like integrate multiple signals into a protein, which is only active if multiple signals are present. You can have multiple effects. You can have branching, such that you can achieve a lot of different actions because of a binding event. Things can be inhibited, things can be amplified, and very often the event that's going to occur in the end is the activation of gene transcription. We'll see on the next slide scaffold proteins, which is a sort of insulation which helps maintain the signaling fidelity and also increase the kinetics. By having the scaffold protein here, the protein kinase 1, which might be promiscuous under other situations, is only going to phosphorylate protein 2, which again would in turn phosphorylate protein 3. Because they're held in close contact, this will occur very rapidly and also minimizes the chance that it's going to interact with other proteins. We talked about the amplification of signaling in the case of the rhodopsin proteins that are present in your retina. This is in the back of the eyeball here. Light is entering through here. It has to pass through the cell body when it gets to the point where you have the rhodopsin molecules. And then the amplification is shown here, that a single photon produces activates about 500 G protein molecules, which will then activate 500 enzymes that are going to create cyclic GMP molecules, about 10 to the 5th. These 10 to the 5th cyclic GMP molecules are going to close about 250 cation channels, and that's going to, this single photon is going to cause changes in 10 to the 6th or 10 to the 7th sodium ions of their location. So that's quite an impressive amplification of a signal. This change in the positions of all these sodium atoms is going to create a change in a membrane potential, which is going to allow an action potential to occur, which will be interpreted by your brain as having seen a photon. So this can all respond to a single photon. Your eye does not see every photon. In general, under the most sensitive conditions, your eyes can see about 1 out of every 3 photons. Having completed talking about the mechanisms of cell signaling, we talked about 3 different types. The simplest being the ion channel coupled receptors, again, where a ligand would bind to a channel, biasing its open versus closed state, and allowing ions to pass through when it's opened. Since we've talked about that, I won't say anything more about them. We talked about G protein coupled receptors, which interact with trimeric G proteins as being a fundamental signaling pathway type. Here we have an example of an inactive trimeric G protein, which it's inactive, it's bound to GDP, not GTP. Upon binding of a ligand to its receptor, which is the 7-transmembrane G protein coupled receptor, this will cause the activation of these trimeric G proteins. GDP will be released, GTP will be bound. This causes dissociation of the alpha subunit from the beta gamma complex, and either of these can go on to produce various phenotypic effects. One of the effects that the book chose to highlight was a beta gamma complex going over to potassium channel and opening this up, allowing potassium to flow out of the cell. When the GTP is hydrolyzed, this is going to inactivate this complex and cause the reassociation of the trimeric G proteins. You'll note that these trimeric G proteins are, again, what I call the balloon on a string model, where they're attached or they're held in close contact with the membrane by these lipid attachments, which are stuck in the membrane and cannot diffuse out. We also spent a little time talking about mechanisms other than the phosphorylation of proteins, again stressing the amino acids serine, threonine, or tyrosine, which have hydroxyl groups, that ways of transmitting signals could be using small molecules like cyclic AMP. In this case, the binding of a ligand has activated an enzyme which will take ATP and cause the cyclic form between the 5' and 3' end, and this molecule is bound by, in this case, protein kinase A, activating it, and then it's going to go on and do downstream effects. Eventually, the cyclic AMP is cleaved, producing adenosine monophosphate, and that will terminate the signal from this molecule. The second type of small molecule that we talked about is an inositol phospholipid, which upon signaling, again, of this G protein coupled receptor, will activate an enzyme called phospholipase C. It will cleave this protein called inositol triphosphate from the diacylglycerol, which remains in the membrane, and both of these molecules are active in the signaling pathway. This will open up a channel which then can get released. The two signals converge on protein kinase C, which binds to the diacylglycerol, and now it's activated for downstream effects. This is just sort of a blow-up of the molecular detail. Again, inositol is a sugar, not that dissimilar from glucose, but it's phosphorylated 3 times. And the diacylglycerol, here you can see the glycerol backbone. Once it's cleaved, you have a free hydroxyl group and 2 fatty acid chains, which are diffusing in the lipid bilayer. A third type of signaling pathway which has a lot of attention because of its role in cancer are receptor tyrosine kinase pathways, which your book refers to as enzyme coupled receptors. But again, of the 3 amino acids that can be phosphorylated, Tyrosine is the one that's phosphorylated most rarely. There are many more examples of phosphorylation of serine and threonine. And the phosphorylation of tyrosine has been reserved somewhat specifically for a particular function, and that is promoting cellular growth. And so that's why it's of interest to cancer, because you want to block cellular growth in cancer. The way these signals are transmitted is that you have a dimeric ligand, which will cause the ligand-induced dimerization of the receptor monomers. This turns on the activity of kinase domains on the cytoplasmic side of the proteins. It will phosphorylate the protein. Now these are active, and then it can achieve the phosphorylation of other target proteins, which will continue to transmit the signal into the interior of the cell. Again, after ligand-induced dimerization, one of the important downstream proteins is a protein called RAS. It's a GTP binding protein. It's a monomeric GTP binding protein as opposed to the trimeric G proteins. Once it is bound by GTP, it's active and it's going to phosphorylate other proteins, specifically this MAP kinase pathway, which has a MAP kinase, and MAP kinase. Your RAS is phosphorylating it. That's going to make it active. It's going to phosphorylate its target. It's going to phosphorylate its target. In all cases, the phosphoryl groups are provided by ATP. They are not transferred from this protein to this protein, but eventually you're changing the activity of proteins, particularly those associated with cell growth. And RAS is a frequently mutated protein in cancers, and what happens here is that it cannot hydrolyze GTP, so it's stuck in the on position even if there's no ligand bound to the receptor and it's not supposed to be firing. It's going to be firing just as if it had received a signal, and this is one of the sources of inappropriate growth of a cancer cell. On this last slide from this section, we highlight the fact that many signaling pathways interact with each other and can produce coordinated effects. Unfortunately, many of these times it also reflects our lack of real understanding in terms of how signals are transmitted. Some of these interactions make sense here. The MAP kinase pathway, which can promote cell division, is also coupled to a protein that goes, a pathway that goes via AKT, which produces cellular growth, meaning the accumulation of mass, which you want to accomplish if you're going to be dividing as a cell. Alright, in the next segment we looked at the cytoskeleton and cells' interactions with the extracellular matrix. In this picture here we can see in green microtubules proceeding radially from the center of the cell. We can see actin forming a band around the periphery. At the leading edges, not pictured, are intermediate filaments. The nucleus is pictured in blue. Cytoskeleton allows cells to generate and resist forces, and it helps organize other structures, including allowing them to move within the cell to accomplish their necessary functions. There are three types of intercellular cytoskeletal elements. The intermediate filaments, which are primarily structural, called intermediate because they're intermediate in size between the larger microtubules and the smaller actin filaments. These provide largely mechanical strength. There are microtubules, which has a lot of dynamic properties. They're responsible for organelle movement, vesicle movement, chromosome movement during mitosis, and then actin filaments, which in addition to contraction are at leading edges of cell shapes and cell movements, and a number of other processes, like we just saw phagocytosis, where it caused the plasma membrane to grow around a bacterium. We'll talk about each of these in turn. Starting off with the intermediate filaments, they are primarily located around the periphery of a cell. This is an epithelial cell, which is the boundary between the outside world and the inside world. You can see that there are points of attachment here, which we'll discuss later, which help strengthen the intermediate filaments both of the cell and also of the connection between the two. There are many different proteins that contribute to intermediate filaments, one of the most common being keratin, which you've heard about in hair or wool, but they all have this basic shape, which is a long helical coil. Then they dimerize, tetramerize, and then 8 tetramers will be wound up in a bundle, giving it a very strong mechanical strength, allowing the cell to withstand mechanical forces. This slide emphasizes the role intermediate filaments play in cell-cell contacts. We can see the large nuclei and intermediate filaments bridging spaces between cells, which serve as reinforcements to give the sheet as a whole mechanical strength in addition to just the cell. There are a number of diseases in which the intermediate filaments that are responsible for providing cell strength are absent or defective, and in these cases you can see sloughing of skin, rupturing of the epithelial layer, which can go on to lead to a large number of infectious and immune diseases. Another role of intermediate filaments is to provide structure for the nucleus. Here, nuclear lamins form a dense mesh network right underneath the nuclear pore. Here's the cytosol, here's the nuclear matrix, and this gives strength to the nucleus. This nuclear lamina dissociates right at the initiation of mitosis, allowing the chromosomes to separate, and then reforms as the two new cells start to reform, and again spontaneously reassembles into the nuclear lamina. One point, these proteins are called lamins, not to be confused with laminins, which are part of the extracellular matrix. The next cytoskeletal fiber is microtubules. They are regarded primarily as the highways of the cell. They emerge from a centrosome in the center and they extend radially in the cell. There are other organelles which serve as nucleation sites for microtubules. We'll see that on the next slide. Microtubules are made up of 2 proteins, alpha and beta tubulin. They form about 13 little filaments that combine to form a single larger filament. They have a polarity, meaning that the beta tubulin is always on the plus end, the alpha tubulin is always on the minus end. The minus end is the one that connects to the centrosome. This is an example of some of the other organizing centers for microtubules. Basal bodies allow cilia to project out into, for instance, the intestinal lumen. The sliding of the microtubules next to each other gives the cilia a wave-like motion, and they're also part of the mitotic spindle, which is crucial for cell division, pushing the cells apart, pushing the chromosomes to the center, and then retracting them at the end of cell division. Each microtubule coming out of this centrosome can be individually regulated. They can grow or shrink dynamically based on a number of regulatory proteins, the amount of GTP present, and the amount of tubulin monomers present. And here we see that tubulin-bounded GTP favors its association, although it can also dissociate, but then with some constant time, GTP will hydrolyze into GDP, and that will promote dissociation. Again, it's just a bias to an equilibrium with slightly favoring growth when GTP is present, slightly favoring dissociation when GDP is present. This slide shows an example of how microtubules can be used to position a nucleus asymmetrically in a cell, which means not in the center but closer to one edge rather than the other. You have microtubules growing out in all directions. Some microtubules will hit the membrane, will push out, will extend. They'll have special capping proteins which will stabilize them so that they don't shrink, and that will produce a mechanical force pushing the nucleus to one side of the cell. This is a key process in the formation of epithelial cells where the nucleus is always closer to the basement membrane rather than the apical side. With regard to organelle transport within a cell, the best example of that is in axons, which have long, slender-like projections called axons, which can extend over macroscopic distances. Vesicles need to be transported from the site of protein synthesis, perhaps to the site of at the axon terminal, and these are transported in vesicles by attaching to motor proteins which hydrolyze ATP to walk along the microtubule. So this is not diffusion, this is active transport. Depending on the motor protein that you interact with will determine your direction. Kinesins going away from the nucleus, dyneins going towards the nucleus. And here that's made clear. Kinesins, dyneins control the direction of transport of microtubule, the minus end being near the nucleus or the centrosome, the plus end being near the periphery of the cell. Actin filaments form many structures within cells. They can also be at microvilli. They can be involved with providing strength and shape to flat cells. They can be at leading edges of cells that are crawling in a particular direction. They also form a contractile ring during cell division, helping to perform cytokinesis. The actin monomer is primarily a single actin molecule that has a polarity with a minus end and a plus end. These red, white, and blue colors are just different instances of the protein, not different proteins. And this filament has the fastest of all the polymerization rates. They can grow very quickly. Unlike microtubules, actin filaments can grow at both ends. There's an ATP end on the plus end and an ADP end, but again, this is just biasing the association-dissociation constants, and actin filaments can grow on both ends. They can form a number of different structures. There can be rods, there can be interconnected networks, there can be meshes. These are all controlled by many, many proteins which interact with actin to create diverse structures. With cell movement, there's two components. One, there's actin polymerization at the leading edge, forcing the cell to protrude here. At the same time, there are contacts between the cell and the ground, treads, if you will, to prevent slipping of the cell, and these are contracted. The integrin proteins, which are making contact with the extracellular matrix, will have their connections broken, allowing the cell to move forward as it crawls towards a new location. Actin, of course, is best known for its role in muscle contraction with the large molecule myosin. You have a unit called a sarcomere, which has actin molecules connected to Z disks. In between there, you have myosin heads, which can crawl along the actins. This portion of the myosin crawls in this direction. This portion of the myosin will crawl in this direction. Since this is not going to stretch, this is going to actually pull these two pieces closer together. Here you can see the distance separating them is smaller and the Z disks have moved. What's rather amazing to think about when we think about how fast our muscles can move is to think that for macroscopic distances, these tiny myosin heads must make thousands and millions of individual steps crawling along the actin, each time pumping ions in and out of the cell. It's rather remarkable that can happen as fast as it can. The hydrolysis of ATP is the way that the cell powers this contraction, but it's perhaps not as many suspected. The force-generating power stroke occurs when ADP has released the inorganic phosphate and then has released itself. That causes this large conformational change, which is the walking of the myosin head on the actin. Once it's made its step, it's in a frozen position attached to the actin. ATP will come in. This will allow it to detach, and as ATP is hydrolyzed, this will allow the head to extend and get ready to begin to take another step. On this slide is an overview of the cell's interactions with other cells and the extracellular matrix. We're going to focus particularly on two types of proteins, cadherins, which mediate cell-cell contacts, and integrins, which mediate cell extracellular matrix contacts. We'll focus first on cadherins, which mediate cell-cell contacts. We'll talk just very briefly about adherens junctions and desmosomes. The difference here, the cadherins are interacting with each other, promoting cell-cell contact. At adherens junctions, they are interacting with actin in a band around the cell, providing mechanical strength. At desmosomes, cadherins interact and in the form of spot welds. This is just a particular point on the cell wall that's connecting to give the epithelial cell layer greater strength. There are many different types of cadherins in the body, and they're specific for different cell types and locations, and they preferentially interact with each other. such that if you take two tissue types and dissociate them into cells, they will reassort themselves into independent units because of the shared cadherin interactions of the particular tissue type. You can also get organization based on the amount of cadherin expression, and you can define an inside and an outside based on the amount of cadherin that's expressed on the surface. The cadherins are always connected to cytoskeletal elements on the inside of the cell, providing a connection between the cell-cell contacts and the intracellular cell structure. The integrin proteins here again are transmembrane proteins. They're connecting to extracellular matrix proteins, laminin and collagen, forming the basement membrane underneath the epithelial cell layer. They're connecting to intermediate filaments on the inside of cells, again, providing structural continuity between the intracellular structures and the extracellular structures. The interaction of integrins with the extracellular matrix are at places called hemidesmosomes because it's only one cell that's involved and it's connecting to the intermediate filaments. Here you have desmosomes where you have two interactions, again mediated by cadherins, but connecting to the intermediate filament network of the cell. Overall, this picture is a good summary of all the cytoskeletal elements and their interconnections, and it'll be useful for your review. And I include this slide simply as a convenient reference point for the most important points on these different cytoskeletal elements. In the next to last section, we talked about the cell cycle, which in essence is you have a cell with a normal diploid set of chromosomes, one copy from your mother, one chromosome from your father. During the cell cycle, the DNA is duplicated. That precedes a mitotic cell division, producing two cells, and the cell cycle will repeat. The modern view of the cell cycle is divided up into four phases. There's the G1 growth or gap phase. There's the S phase where DNA is synthesized, a second gap or growth phase, and then the M phase where mitosis occurs. And despite prior emphasis, the actual amount of time the cells are doing mitosis is very short compared to the interphase, all these other phases where most of the biology is going on. This is the old view of the cell cycle. You have this interphase, which even the name suggests not much is happening. It's like an intermission, and then you have all the action of the M phase mitosis. You have IPMAT as the acronym: interphase, prophase, prometaphase, metaphase being a key point when the chromosomes are lined up in the middle, anaphase when they start to separate, telophase when nuclei start to reform, and cytokinesis, which is division of the cell cytoplasm. When we combine the two, we see that interphase is the G1, the S, and the G2 periods, and the M phase is, again, the mitotic phase. Part of the reason for these two different views of the cell cycles, earlier on, we only had microscopes and most of the information was visual, and this is the only time of the cell cycle where interesting visual things were occurring. And when we started to switch over to molecular biology as a primary tool, we were able to decipher all these other molecular events that were occurring during the interphase. With progression from one stage of the cell cycle to the next, there's the idea of checkpoints. We're going to talk about three of these. One of the important ones is the start checkpoint, and this is when the cell, if the environment is favorable and the cell is supposed to grow, it will proceed into the S phase, and this is a committed step towards pursuing the entire cell cycle. There is a checkpoint right before the M phase begins where you're going to check, is all the DNA replicated, are all mutations fixed, and then about halfway through the mitosis stage you have the metaphase to anaphase transition. This is when the chromosomes in the middle are going to start to get pulled apart, and there's going to be a tension test to make sure that chromosomes are attached to the mitotic spindles properly so that each cell will get a full complement of chromosomes. If these checkpoint conditions are not met, then the cell will pause and it will spend some amount of time trying to accomplish the task, whatever is needed, before it proceeds on with the cell cycle. If things get too bad, the cell may choose to commit cellular suicide or apoptosis. The other point to mention is that there is a specialized resting state called G0, which we sort of view as a parking lot on the side of the cell cycle where cells can hang out. They can be metabolically active, but they're not actively cycling. Before they continue cell division, they will go back into the G1 phase and then re-enter the cell cycle. And again, there are more DNA checkpoints or cell cycle checkpoints, but again, we're just going to focus on these three. The committed point to entering the cell cycle, a DNA integrity check, mutation check, a duplication check, and then a proper attachment check for cell division. Progression from one phase of the cell cycle to the next, for instance entering into the M phase of mitosis, depends on a series of molecules called cyclins. These cyclins have concentrations which build up over the course of the cell cycle, and when they hit a critical mass, they will create the molecular changes necessary for entry into that stage of the cell cycle. There are S cyclins for entry into the S phase. There are G1, G2 cyclins for entry into those phases. The way that cyclins will then initiate the changes is they will activate compound proteins that they're partners with. These are the CDKs. There are, again, specific CDKs for each phase of the cell cycles, and these are kinases that themselves are activated by phosphorylation, and these will go on to trigger changes in transcription factors so that you'll start to synthesize the genes needed for the next stage of the cell cycle. The details about the inhibitory phosphate and an activating phosphate are not needed for this level of the class. Just the knowledge that cyclins, when they build up to a certain level, they'll activate the CDKs, which will then go on to activate transcription factors for progression into the next stage of the cell cycle. And here we see the way the cyclins can build up the G1S cyclin, and then the S cyclin, and then the M cyclin. And these cyclins and cyclin-dependent kinases affect the levels of each other. One unique thing about the M cyclin, in conjunction with the metaphase to anaphase checkpoint, is that to exit mitosis, to proceed past this checkpoint, you must physically degrade the M cyclin. If you don't degrade these other cyclins, for instance, if G1S were to persist at a high level here, you could still enter into the S phase because of the rise of the S cyclin. That is not the case for mitosis. You have to destroy the M cyclin to exit mitosis and to begin with the next set of G1 cell cycles stages. One of the key proteins for regulating the cell cycle is this protein called 53, often referred to as the guardian of the genome. P53 is constantly synthesized and constantly degraded in the cell, so it's always maintained at low levels, but with high rates of synthesis and high rates of degradation. If the cell experiences DNA damage, all of a sudden this P53 is stabilized, and P53 is continued to be synthesized, but now it's not being degraded, so the level of P53 will rise. P53 will bind to DNA and start to transcribe a series of genes, like the P21 gene, which can inactivate one of the cyclin-dependent kinases, so putting a halt on the cell cycle progression. The cell cycle arrest is an attempt for the cell to take enough time to try and repair the DNA damage that the cell is sensing. If the amount of DNA damage overwhelms the DNA repair capacity, cell cycle arrest, or the cell will undergo apoptosis, committing cellular suicide so that a damaged DNA genome is not propagated. This decision of cell cycle arrest or apoptosis is controlled by the protein P53. So here we see an overview of the steps of mitosis, and I want to highlight the acronym IPMAT, although you could argue whether there should be two P's for prophase and prometaphase. I just point that out so you can remember the relative stages of the mitosis, and the way to remember it is metaphase. The middle phase is when the chromosomes are in the middle. They're lined up in the metaphase plate, and so that you can, when someone asks you, well, when does the nucleus degrade? Well, you could reason that the nucleus has to be degraded before you can align the chromosomes in the middle of the cell, so that you would know that that might be something that happens during prophase or prometaphase. Similarly, if you remember the acronym IPMAT and say, what is the phase where the chromosomes start to separate, you might immediately be able to think of anaphase as a stage where the chromosomes are separating, telophase where the nucleus starts to reform, and cytokinesis, which is the cleavage of the cellular cytoplasm. Now this is mitotic overview. So these are the various phases: chromosome condensation, nuclear breakdown, attachment of chromosomes to the mitotic spindle, alignment at the center of the cell, separation of the chromosomes to the two spindle poles, reformation of the nuclear pore, the beginning of separation of the cells, cytokinesis, the cleavage of the cytoplasm, and that concludes that. You can review those on your own. For the mitotic spindle, you have a centrosome, which again is the organizing principle for the microtubules, which gets replicated during the S phase, so that you have mitotic spindle where you have microtubules pushing these two sort of organizing centers further and further apart. They'll organize or orient themselves on either side of the nucleus, and the microtubules that are interacting here are pushing against each other. The microtubules on the other side are positioning themselves against the cell wall. As they're shooting microtubules out, some of them will hit the kinetochores, the centromeres of the chromosomes, and if it happens to be here, it'll get hit from this side statistically more often. It will push the chromosomes to the middle. Eventually, a microtubule from the other side will hit this centromere from the other side and push it back into the middle of the cell at the metaphase plate. The interpolar microtubules position the centrosomes far apart from each other, and the astromicrotubules position the centrosome with respect to the cell boundary on the far side of the cell. Showing some of the details of the orientations here, we have dyneins, which are going to walk towards the negative end, pull the membranes in close to the side of the centrosome. We have special types of kinesins, which have multiple heads, which will be walking on the positive sides in both directions, again pushing these centrosomes apart. Other kinesins pushing this chromosome out towards the other centrosome, towards the middle of the cell. And then during anaphase, you have some dyneins that are carrying the chromosomes walking along the microtubules in towards the centrosomes. At the same time, you have these interpolar microtubules, which are pushing against each other, pushing these centrosomes further and further apart. But no need to distinguish between A and B parts of anaphase. The metaphase to anaphase checkpoint, which tests the association of the chromosomes with each of the mitotic spindles, looks for a configuration where there is equal tension on both sides of the chromosome. If there is an imbalance of forces, this will cause cell cycle arrest and or apoptosis, the point here being that the cell wants to make sure that if it's going to divide, that each daughter cell gets a copy of the sister chromatid for each particular chromosome. Mitogens are a particular class of ligands which will trigger cell division. They will do this by inactivating a protein called RB, or the retinoblastoma protein. This is a brake on the cell cycle. This is a tumor suppressor. When it gets inactivated by phosphorylation, now genes are transcribed that is going to pass the start checkpoint and allow for cell cycle and cell division. There are growth factors that act primarily by receptor tyrosine kinases, and this is an example of growth. These are both one cell, but obviously this neuron is much larger than this lymphocyte. So these growth factors can promote protein synthesis, cellular growth, lipid synthesis, and again there will be some interconnection with cell division pathways, so some extracellular ligands can act both as growth factors and mitogens. There's not a real clean division between those two. Then there are survival factors, which are secreted in here by target cells to neurons, and this is a planned process of the body. It wants to regulate to make sure that there's only a single neuron attached to each of these target cells. But it also wants to make sure that there's no target cell that doesn't have one nerve attached to it. So that's why initially it starts out with a surplus and then only keeps the ones that it needs via this survival mechanism. Another example of apoptosis programmed is the limb bud, which initially is one appendage, and then the tissues, the cells in between these digits will commit cell suicide on purpose, and you can see the emergence of digits there. In contrast to necrosis, apoptosis is a very orderly cell death. You see this blebbing, these vacuoles being formed. You'll see it being engulfed by cells of the immune system and digested and recycled for use by the body. Whereas necrosis is something that occurs due to physical damage or abrupt lack of ATP, which can happen because of blood supply or oxygen supply. It's a rather messy thing, leading to large inflammation. The cell will lyse, burst, and release its contents to the extracellular fluid versus, again, sort of this sort of controlled method of cellular disposal. There are two ways of triggering apoptosis: the extrinsic pathway, which is a type of immune cell called a killer lymphocyte, which is going to interact with a death receptor. As drawn, this is a receptor that needs to have multiple interactions, and the ligand has multiple parts that's going to interact with the ligand. It's going to set a set of proteases called caspases will activate each other and will start to chew up the nuclei, the chromosomes, and the proteins part of the cell. Probably the more studied and statistically more important pathway is the intrinsic pathway of apoptosis, and this is when cellular damage is detected, for instance, DNA damage. Cytochrome C, which sits there on the inner mitochondrial membrane, there are proteins which will cleave that lipid molecule off the cytochrome C. Pores will form in the outer mitochondrial membrane. Cytochrome C will escape into the cytosol, and this will be the trigger of the activation of the caspases, which will then go ahead and start the digestion process of nucleic acids and proteins that will eventually lead to apoptosis. This use of cytochrome C as a signaling molecule for apoptosis and an electron transfer protein in oxidative phosphorylation is an example of pleiotropy, which is the word that means that a single gene can have more than one function. And I also want to highlight the critical importance of this pore. And on the next slide, I will show you a protein called BCL2. And this is an example of how a survival factor works. The survival factor will bind to a ligand. They'll have the intermediate activation of different proteins. You'll make the BCL2 protein, which will block pore formation and block apoptosis, keeping the cytochrome C inside the mitochondria. And in the last section of the course, we talked about meiosis, Mendelian genetics, and modern genetics. Start here on this slide comparing and contrasting mitosis versus meiosis. Mitosis produces 2 cells, meiosis produces 4 cells. The goal of mitosis is to produce cells that are identical, whereas meiosis is meant to contribute to genetic diversity. All the cells are different. One of the mechanisms of creating that diversity is crossing over between chromosomes, where in mitosis there's no crossing over. Mitosis occurs in all cells. Meiosis only occurs in the gonads, the cells involved with sexual reproduction. And the main mechanistic difference between the two lies in metaphase 1. The chromosomes align differently, and we'll see that leads to a difference in terms of what gets separated in anaphase 1 versus anaphase 2. So there's 2 ways in which genetic diversity is created by meiosis. Here we have 3 pairs of homologous chromosomes. We have the copies from your mother, the copies from your father, and because these chromosomes are going to sort individually, they're at first order 8 different types of progeny cells that you can make that's going to use different combinations of the green, the black, the red, the blue, the yellow chromosome. Again, this 8 is just 2 to the third. But this in fact never happens because you don't transmit one of your parent's chromosomes in total to your offspring. What happens is there's going to be crossing over, and each chromosome that you're going to pass on will be part maternal and part paternal. So these are creating chromosomes that have never appeared before, that are completely unique to your cells. With a normal amount of genetic material are referred to as diploid. You have 2 copies of each chromosome. During meiosis, you have the halving of genetic material, and so the eggs and sperms are referred to as haploid. And then when you have fertilization of an egg by a sperm, that you resume the diploid status of the zygote, which can then undergo mitosis and create an entirely new organism based on its new genomic inheritance. In terms of mutations that get passed on to your offspring, it's only mutations that occur in the germline cells that affect your offspring. And in your adult body, in your somatic cells, a mutation occurring here may be very deleterious to your health, but it will not be passed on to your offspring. On the right here we have the cellular division mitosis, which we've just seen. Here we have duplicated condensed chromosomes. Here we have metaphase where they're lined up, and these are the duplicated chromosomes themselves. You're going to separate the sister chromatids, the duplicated material, and if this were chromosome 1, this would be copy 1, your maternal copy of chromosome 1, and this would be your paternal copy of chromosome 1. Each daughter cell has 1 copy of each of the chromosomes, genetically identical to the parent cell. In meiosis, you have an extracell division. You have chromosomes lining up, separation of chromosomes, and an intermediate stage where you have 2 cells. Then the chromosomes will line up again. You'll have a further cell division, and now you're going to have 4 cells at the end. And it's here at metaphase where things are different. You have homologous chromosomes lining up next to each other, and they're going to cross over before they get separated. Next slide shows this in more detail. Here's mitosis. You have each duplicated chromosome taking its own slot. Both of them, all sister chromatids, are connected to opposite centrosomes. Here you have homologous chromosomes, so that you have the maternal copy and the paternal copy, which are duplicated. They are connected to opposite spindle poles. Before they separate, they will cross over, and all 4 sister chromatids will exchange some genetic material. But this is metaphase 1 in meiosis. So anaphase 1 is going to separate the homologous chromosomes after they've been crossed over. And then if we look at this one right here, these are sister chromatids. In the second anaphase, they're going to pull on the sister chromatids and separate them during anaphase 2. So then we talked about Mendel's study of genetics. He happened to look at 7 traits of plants. They were all simple Mendelian traits, meaning that there was no blending of phenotypes. For instance, flowers are either purple and white. There's no light purple, which would be a blended trait if you had 1 copy of each chromosome. There's, in each case, 1 phenotype was dominant and a second one was recessive. Looking at P shape, P color, plant color, the location of flowers, the shape of the pod, the color of the pod, and the size of the plant. Pea plants have 7 chromosomes, and these 7 traits happen to be 1 on each chromosome, which is a remarkable coincidence and has raised some eyebrows. But nevertheless, Mendel had tremendous insight into the inheritance of phenotypes. So now, had the different traits not been on separate chromosomes, many traits that even appear on the same chromosome can be inherited independently. For instance, the traits A, the gene at A, and the gene at locus D, or position D, would be inherited independently because there's always a chance of 1 or more crossing overs occurring between those 2. If genes are very close to each other, it's unlikely that a crossing over will happen between them, and they will be inherited together. So A, and you won't see any big A, little b. You'll only see big A, big B, or little a, little b. These regions of chromosomes which have never been crossed over in between are what we will refer to as haplotype blocks. So Mendel looked at particular types of breeding experiments. He only worked with pure breeding plants, which the first ones are referred to as the P1 generation. Then there's the F1 generation and the F2 generation. You get a pure breeding strain by always breeding green round with green round for many, many generations, and eventually everything becomes homozygous when you do that. So when he bred yellow-seeded pea plants with green-seeded pea plants, he found that the initial offspring were 100% yellow. Not random, not an intermediate color. This would later be a clear indication of dominance, which was not a well-defined concept at the time. Then if he took the F1 generation, bred them with each other, the F2 generation, the grand daughters and sons of the P1 generation, he always got the ratio of 3:1 of yellow to green. And this occurred regardless of the trait he was looking at, whether it was the shape of the pea, the color of the flowers, the height of the plant. It didn't matter. He always observed a 3:1 ratio in this scenario. The way that you get to the scenario is you have to look at the genotype and do a genetic cross, which is done here on this Punnett square. And you have to know or get some information that if you say it's a pure breeding strain, you can assume that it's homozygous, and so that there's the dominant phenotype is typically represented by a capital letter, the recessive phenotype is a small letter, and that you know that this plant can only give a large Y chromosome, this plant can only give a small Y chromosome, so all the F1s are going to be big Y, little y. To calculate the F2s, you will cross the genotypes of two different F1 organisms, F1 organism 1, F1 plant 2, and then you do the simple matrix calculation that 25% of the F2 generation is going to be big Y, 50% is going to be big Y, little y, and 25% is going to be little y, little y. So you would report that in terms of phenotype, it's 3:1 yellow to green, but in terms of genotype, it's 1:2:1, homozygous wild type, heterozygous, and homozygous recessive. This is an important skill to be able to do. Things can get more complicated. You can look at the inheritance of two traits at once, pea color and pea shape, yellow and smooth and green and wrinkled. The F1 will be entirely yellow and smooth, those being dominant. This would be the genotype of all of the offspring because it's going to inherit a big Y and a big R from the yellow pea, small y and small r from the green pea. And then you're going to cross this as slightly more complicated. The phenotypes in this case will be 9:3 to 3:1, with 9/16 being showing the dominant traits, 1/16 the smallest amount showing both the recessive traits, and 3/16 of each showing one recessive trait and one dominant trait. I won't go through the genotype proportions here, but they're easy calculated. You can see that a quarter of the pea offsprings are heterozygous for both conditions. There is one case of being homozygous dominant for both conditions. There are two ways of being dominant for one color, one trait, and heterozygous for the other. But this all results from a genetic cross. I'll remind you that the F1 generation was entirely heterozygous, so it had one Y, one big Y, a little y, big R, little r, so that you figure out the different combinations you can have, and they occur with equal frequency: big Y, big R, big Y, little r, little y, big R, and little y, little r. And just to highlight, these results give you exactly the same results when you were considering one trait at a time. Remember, that was a 3:1 ratio. Here you can see a 3:1 ratio of color, and you can also see a 3:1 ratio of wrinkle or shape, in each case 4 out of the 16 have that trait. It's only that these traits are inherited separately that creates the complexity. In terms of modern genetics and anticipating our conversations about cancer, there are different kinds of mutations. If this is a wild-type protein that folds properly and has, a certain activity, there can be loss-of-function mutations. Here's a point mutation which destroys activity. There's a truncation so that this part of the protein doesn't even exist, or the gene deletion. There can be gain-of-function mutations, which are less common, but RAS was an example in the signaling pathway where it's always on. And then there can be conditional mutations where activities don't work at certain temperatures but work at others. Then in terms of identifying which genes are responsible for a particular disease or a particular trait, linkage disequilibrium is one of the methods that has been used quite frequently in somewhat recent history. These studies require large extended pedigrees, large families, and you gain extra information because of the inherited relationships between the different generations. You also make the assumption that everybody's disease is occurring because of the same mutations. When you look in populations as cross-sectional without related to their family tree, you know that different mutations can cause similar appearing diseases, so that can make tracking things down a little more difficult. But because you're dealing with smaller numbers of people and smaller numbers of crossing over events during meiosis, if you find a region of a chromosome which is responsible for the disease, it's ordinarily a larger portion than if you're doing a cross-sectional study with thousands of people. And in these types of pedigrees, squares are males, circles are females, blue is affected, and the white color is healthy. In association studies, we're going to sequence lots of portions of the genome, and we're just going to do a statistical analysis. Is there any genetic element that appears more often in the diseased population than the healthy population? In this example, the yellow was created with the idea of saying that is what's responsible for the disease. Here there's a healthy person which has the susceptibility for the disease but has not yet expressed the disease, and that happens all the time in modern genetics. Nowadays, association studies are requiring many tens of thousands to 100,000s of people to try and fish out the genetic associations with phenotypes. This was not anticipated. People felt like the genetic causes of these diseases would have been more readily apparent. That's one of the challenges for modern genetics. When we're sequencing different parts of the genome in all those thousands of people, we're not sequencing the whole genomes. We know that the chromosomes come in blocks of haplotypes, and that if we know where the chromosome has never been crossed over in all of human history, so that the block is inherited as a whole, if we genotype one SNP, one single nucleotide polymorphism, that's a base pair change. You can think of SNPs as common mutations. Any single base pair change that is present in more than 1% of the population might be called a SNP. If it's rarer than 1% of the population, then it's typically called a mutation. And by sequencing this one base, we'll know what flavor of allele or haplotype a person has at that portion of the chromosome to a good approximation. So it's a very efficient way of querying genomes. So in this slide, we depict chromosomes from multiple individuals, 10, 5 healthy and 5 affected. And what the colored lines indicate are SNPs, single bases that vary in the population. And what you notice is that there's a region here where a stretch of the chromosome only comes in 2 varieties. It can come in all red or all blue. The fact that these 2 varieties segregate with the phenotype, whether it's affected or not, indicates that there is a disease-causing gene somewhere in this haplotype block. Now, although there's 4 SNPs here, you would only have to genotype one of these SNPs to know what all 4 of them are, because as we can see, there are only 2 varieties of chromosome here. And although it's less easy to see, there is also another haplotype block here where there are only a few varieties. For instance, red, red, blue, blue, blue. You can see that same pattern appears over here, red, red, blue, blue, blue. So this is a portion of a chromosome that is being inherited together, another haplotype block. So anyway, we see in the dark gray haplotype block that there is a association between particular SNPs and disease, and so that means there is a gene somewhere in here that is causing the disease. It may not be one of those SNPs. It could be, but it usually isn't. And the way haplotype blocks are discovered is when people genotype many different SNPs along a chromosome and see which ones are correlated with each other. Darker colors mean that SNPs are correlated. For instance, this SNP has a high correlation with this SNP right here, so this region of the chromosome is a haplotype block, meaning chunks of chromosome that frequently get inherited together. And then finally, when we look at complex traits, not Mendelian traits, we see that there is shared genetic and environmental influence in many of these cases, that there's a spectrum, some things like multiple sclerosis, which depends on infections that you may have experienced, or cancers, strong environmental influence whether you smoke or not, as opposed to things that are more genetically based, such as psoriasis, and all the Mendelian traits would be way over here. Those Mendelian traits were entirely determined by the genes and not the environment. So that concludes the review for midterm 2. Thanks for hanging in there. I apologize for my voice. There's nothing I can do about that. I wish you luck on the second midterm. Please come to me if there are any questions that you want to discuss.